Hello students, with me Miss Yessica. So today, you're going to continue our lesson and it's about the blood. So last week, we have learned about the transport systems in mammals, which I have briefly explained about it. Today, we're going to talk about the blood. And my question is, why you should learn about the blood and what makes the blood important? So basically, we know that blood is the element that was carried in the transport systems. So that's why you should learn about the blood. In this slide, you can see the pictures of the blood that are already separated by the centrifuge. So after the separation, we know the composition of the blood that consists of 55% of the blood, they are the plasma, which is a variety of inorganic and organic molecules dissolved in the water, and it represents by the color of yellow in the picture. While 55% of the blood that consists of the red blood cells, white blood cells, and platelets. So in this picture, you can see that there's a red color there. The red color is the erythrocytes or the red blood cells, which is around 55% of total blood. Well, for the leukocytes or the white blood cells and the platelets, they are less than 1% of total blood, which represent by the white color in the picture. After we know the composition of the blood, next, what is the function of the blood? So blood that has three functions, which are for the transport, for the defense, and for the regulation. The first one is for the transport. So blood is used to transport the oxygens from the lungs, nutrients from the digestive tracts, hormone from all parts of the body. It also used to exchange with the carbon dioxide and the waste materials. The second one is the defense. So your blood used to defend your body against the invasions of the pathogens. It can be the virus, it can be the bacteria, it can be the pathogens, fungi, and many more. It also used for the blood clotting. The third one is the regulations. So your blood used to regulate your body temperatures. So blood makes sure that your body temperatures is not too high and also not too low, because if it's too high or too low, it could damage your body. It also used to regulate the body cells or the water balance, and also the body pH. Why the body pH is important? So if your blood is too acid, it means that your body lacks of the oxygens. And this one, if this is the case, it could damage the functioning of your body organs. Plasma. So plasma is the liquid portions of the blood, and 92% of it is water. 8% of it consists of various salts or ions and organic molecules. Salt is used to maintain the pH. Organic molecules, such as glucose and amino acids, it used as the nutrients. Urea, it's the nitrogenous waste. And the other one are the large organic molecules, which consist of hormones and plasma proteins. So, as blood flows through the capillaries within the tissues, some of the plasma leaks out through the gaps between the cells in the walls of the capillary, and they seeps into the spaces between the cells of the tissues. Almost 1/6 of your body consists of the spaces between your cells. These spaces are filled with this leaked plasma, which is known as the tissue fluid. So, the tissue fluid is almost identical in compositions to blood plasma. However, it contains far fewer protein molecules than the blood plasma, as those molecules are too large to escape easily through the tiny holes in the capillary endothelium. Red blood cells are much too large to pass through, so this tissue fluid does not contain the red blood cells. But some of white blood cells, they can squish through and move freely around in this tissue fluid. So from that explanations, we know that it's true that the tissue fluid contains fewer protein molecules than the blood plasma. Blood is composed of the cells floating in a pale yellow liquid called plasma. Blood plasma is mostly water, with a variety of substances dissolved in it. The cells include the nutrients such as the glucose and waste products such as urea that are being transported from one place to another in the body. So solids also include the protein molecules. They are called the plasma proteins that remain in the blood all the time. In 100 milliliter plasma, it contains around 7.6 gram proteins. And there are three primary plasma proteins, which are albumins, globulins, and fibrinogens. Albumins makes up around 60% of the plasma proteins, and it used to regulate the osmotic pressures of the plasma. The globulins it used as the antibodies, and the fibrinogen it used for the blood clotting. The table shows the sizes of the molecules of some of the substances in blood plasma and the relative ease in which they pass from capillaries into the tissue fluid. So, permeability refers to the ease in which molecules cross the biological membranes. As you can see, as the relative molecular mass increasing, the permeability will also decrease. It's much difficult for the bigger substances to cross the capillary. The volume of the fluid which leaves the capillary to form the tissue fluid is the result of two opposing pressures. Particularly at the arterial end of the capillary bed, the blood pressures inside the capillary is enough to push fluid out into the tissue. However, we have seen that water moves by the osmosis from the regions of low solute concentration or high water potential to the regions of high solute concentration or low water potential. Since tissue fluid lacks of the high concentrations of proteins that exist in plasma, the imbalance leads to the osmotic movement of water back into the capillaries from the tissue fluid. The net result of this competing process is that the fluid tends to flow out of the capillaries into the tissue fluid at the arterial end of a capillary bed and into capillaries from tissue fluid near the venous end of the capillary bed. Overall, however, rather more fluid flows out of the capillaries than into them, so that there is a net loss of fluid from the blood as it flows through the capillary bed. If the blood pressure is too high, too much fluid is forced out of the capillaries and may accumulate in the tissues. This build-up the fluid is called edema. One of the roles of the arterioles is to reduce the pressures of the blood that enters the arterioles in order to avoid this. Tissue fluid forms the immediate environment of each individual body cells. It is through the tissue fluid that exchanges of material between the cells and the blood occurs. Within our bodies, many processes take place to maintain the compositions of the tissue fluid at the constant level to provide an optimum environment in which the cells can work. This process contributes to the overall process of the homeostasis, that is, the maintenance of the constant internal environment, and include the regulations of the glucose concentrations, water, pH, metabolic wastes, and temperature. Lymph. About 90% of the fluid that leaks from the capillaries eventually seeps back into them. The remaining 10% is collected up and returned to the blood systems by the means of the series of tubes of lymph vessels or the lymphatics. So the lymphatics are the tiny, blind-ending vessels which are found in almost all tissues of the body. Lymphatics contain the tiny valves which allow the tissue fluid to flow in but stop it from the leaking out. The lymphatic systems. The lymphatic systems consist of lymphatic vessels and lymphatic organs. So, what is the function of the lymphatic systems? First, the lymphatic capillaries takes up excess tissue fluid and returns it to the bloodstream. Second, the lacteals receive the lipoproteins at the intestinal villi and transport them to the bloodstream. The third, the lymphatic systems helps to defend your body against the disease. The valves in the lymph vessels walls are wide enough to allow large protein molecules to pass through. This is very important because such molecules are too big to get into the blood capillaries and so cannot be taken away by the blood. If your lymphatics did not take away the proteins in the tissue fluid between your cells, you could die within 24 hours. If the protein concentrations and the rate of the loss from the plasma are not in balance with the concentrations and the rate of loss from the tissue fluid, edema may result. The fluid inside the lymphatics is called lymph. Lymph is virtually identical to the tissue fluid. It has a different name, more because it is in a different place than because it is different in composition. In some tissues, the tissue fluid and therefore the lymph is rather different from that in other tissues. For example, the tissue fluid and the lymph in the liver have particularly high concentrations of proteins. High concentrations of lipids are found in the lymph in the wall of small intestine shortly after a meal. In here, the lymphatics are found in each phylus where they absorb lipids from the digestive tract. Lymphatics join up to form the larger lymph vessels that gradually transport the lymph back to the large veins that run just beneath the collarbone, the subclavian veins. As in the veins, the movement of the fluid along the lymphatics is largely caused by the contractions of the muscles around the vessels and kept going in the right directions by the valves. Lymph vessels also have smooth muscles in their walls, which can contract to push the lymph along. Lymph flow is very slow. This contrasts with the flow rate of the blood. At intervals along the lymph vessels, there are lymph nodes. These are involved in the protection against the disease. Bacteria and other unwanted particles are removed from the lymph by some types of white blood cells as the lymph vessels through a node, while other white blood cells within the nodes secrete antibodies. Red blood cells, they are also called as erythrocytes, which simply means red cells. The red color is caused by the pigment hemoglobin, which is a globular protein. The main function of the hemoglobin is to transport the oxygens from the lungs to the respiring tissues. So the red blood cells, they are small. They have a biconcave disc that lacks of nucleus when measured. They also have a great quantity. They are about 4 to 6 million per cubic millimeter of the whole blood. A person's first red blood cells are formed in the liver, while still a fetus inside the uterus. By the time a baby is born, the liver has stopped manufacturing the red blood cells. This function has been taken over by the bone marrow. This continues at first in the long bones such as the humerus and femur, and then increasingly in the skull, ribs, pelvis, and vertebrae throughout the life. Red blood cells, they live only for 120 days. Around 2 million of red blood cells are destroyed per minute. Erythropoietin is a hormone produced primarily by the kidneys, and it plays a key role in the productions of the red blood cells by speeds up the maturations. The red blood cells, they do not live for long. Their membranes become more and more fragile and eventually rupture with some tight spots in the circulatory systems, often inside the spleens. So we know that the red blood cells, they contain of hemoglobin. So the hemoglobin will be distracted into the heme and globin. Globin will be converted into amino acids, while the heme will be converted into the bile pigments, which are bilirubin and biliverdin. The structures of a red blood cells is unusual in several ways. First, red blood cells are shaped like the biconcave discs. The dent in each side of the red blood cells increases the amount of surface area in relation to the volume of the cells, giving it a large surface area to the volume ratio. This large surface area means that the oxygens can diffuse quickly into or out of the cells. Second, the red blood cells are very small. The diameter of the human red blood cells is about 7 micrometer, compared to the diameter of an average liver cells of 40 micrometer. This small size means that no hemoglobin molecule within the cells is very far from the cell surface membrane, and the hemoglobin molecule can therefore quickly exchange the oxygens with the fluid outside the cells. It also means that the capillaries can be only 7 micrometer wide and still allow the red blood cells to squeeze through them, so bringing the oxygens as close as possible to the cells which require it. The third, the red blood cells are very flexible. Some capillaries are even narrower than the diameter of the red blood cells. The cells are able to deform so that they can pass through these vessels. This is possible because the cells have a specialized cytoskeleton made up of the mass-like network of protein fibers that allows them to be squashed into different shapes, but then springs back to produce the normal biconcave shape. The last one, the red blood cells have no nucleus, no mitochondria, and no endoplasmic reticulum. The lack of these organelles means that there is more room for the hemoglobin, so maximizing the amount of oxygens which can be carried by each red blood cells. Platelets, they are also called as thrombocytes. They are a component of the blood whose function, along with the coagulation factors, is to react to bleeding from the blood vessels injury by clumping, thereby initiating the blood clot. So the platelets, they result from the fragmentations of the certain large cells, the megakaryocytes, and they are produced at the rates of 200 billion a day. Blood contains around 150,000 to 300,000 per cubic millimeter. And then the next is hemophilia. Okay, have you heard about hemophilia? Okay, if you haven't, hemophilia is inherited clotting disorder due to deficiency clotting factors. How is the stages of the blood clotting? So when there is a break occurs in the blood vessels, the damaged tissue will release the thromboplastins, which is a blood clotting factors, and this initiates a series of reactions involving several clotting factors and the calcium ions, which leads to the production of the prothrombin activator. The prothrombin activators will convert the prothrombin to become the thrombin, and the thrombin acts as an enzyme that severs two short amino acid chains from each fibrinogen molecule. And this fibrinogen molecule will form the threads of the fibrin, which provide the framework for the clot. So this is how the blood clotting process occurs. Abnormal hemoglobin. So there are two inherited disorders which has the abnormal hemoglobin. They are thalassemia and sickle cell anemia. So thalassemia is the inability to produce adequate amounts of alpha and beta chains of hemoglobin. Rate of red blood cells production is slowed, and the mature red blood cells are fragile and short-lived. People with thalassemia should undergo periodically transfusions. The next is the sickle cell anemia. Sickle cell anemia caused by the mutations of the amino acid sequence of beta chains of hemoglobin molecules. So basically, people with the blood that has normal hemoglobin, and red blood cells, they contain abundant of oxygens. While people with sickle cell anemia, their red blood cells are fragile, easily to match, stiff and curved, so that's why they are defective to bond to the oxygens. White blood cells are sometimes known as the leukocytes, which just means the white cells. So the white blood cells, they are larger than red blood cells. They have a nucleus, they lack of hemoglobin, and they are translucent. The white blood cells, they are around 5,000 to 11,000 per cubic millimeter, and they are found in the tissue fluid and the limb. And they also used to fight infections by the hemostasis and immunity. They derive from the stem cells in the red bone marrow. So there's a process of making leukocytes, which we call as the leukopoiesis. They are stimulated by the various colony stimulating factors, which are the hormones produced by the major white blood cells. And there are two types of white blood cells. They are granular and agranular. So the difference are the granular, they have granules in the cytoplasm surrounding the nucleus, while the agranular, they don't have the granules in their cytoplasms. So I mean with the granules here are the enzymes and the proteins, which helps the white blood cells to defend the body, your body. The white blood cells are also made in the bone marrow, but they are easily to distinguish from the red blood cells in a blood sample because first, the white blood cells all have a nucleus, although the shape of these varies in different types of white cells. Second, most white blood cells are larger than the red blood cells, although one type, the lymphocytes, may be slightly smaller. The third, the white blood cells are either spherical or irregular in shape, never looking like a biconcave disc. Granular leukocytes. So there are three types of granular leukocytes, which are the neutrophils, eosinophils, and basophils. So for the neutrophils, they are most abundant. They have a multi-lobe nucleus joined by the nuclear threads or the polymorphonuclear. They engulf the pathogens during the phagocytosis. The second one is the eosinophils. So they have the bilobe nucleus. They take up the eosins, which is the red color in the picture. And they increase the number of the parasitic worm infections and allergic reactions. The third one, the basophils. So basophils have a U-shape and or lobe nucleus, and they take up the basic stains like the dark blue color. So together with the mast cells, they will release the histamine associated with the allergic reactions. Granular leukocytes. So there are two granular leukocytes, which are the monocytes and the lymphocytes. So the monocytes, they are the largest of the white blood cells. So after taking up residence in the tissue, they will differentiate into larger macrophages. Macrophages phagocytes pathogens, old cells, and cellular debris, stimulate other lymphocytes to defend the body. So for the lymphocyte, there are two types of lymphocyte, B lymphocytes and T lymphocytes. So what is the difference between the B lymphocytes and T lymphocytes? The B lymphocytes produce the antibodies by combining with the antigens to destroy the target pathogens. While for the T lymphocyte, it just directly destroy any cells that has the foreign antigens. And there is a disease called the leukemia. Leukemia is a type of cancer and abnormally large number of immature white blood cells that fill the red bone marrow and prevent the red blood cells to develop. So there are many different kinds of white blood cells with a wide variety of functions, although All are concerned with fighting disease. They can be divided into main groups. Phagocytes are the cells that destroy invading microorganisms by phagocytosis. The commonest type of phagocyte can be recognized by the lobe nuclei and granular cytoplasms. The monocytes are also phagocytes. So the lymphocytes also destroy the microorganisms, but not by phagocytosis. Some of them secrete chemicals called the antibodies, which attach to and destroy the invading cells. There are different types of lymphocytes which act in different ways, though they all look the same. Their activities have mentioned that before. So the lymphocytes are smaller than most phagocytes and they have a large round nucleus and only a small amount of cytoplasm. That's all for today. Thank you for your attentions. Goodbye.